Before we begin, I have a couple quick announcements. Please mute your microphone and keep it muted during the program. This will ensure that everyone can hear our presenters. We're going to leave ample time for a Q&A after the presentations, so please hold your questions until then. And lastly, I wanted to let you know that we're going to be recording this program and we plan to make it available via the college's YouTube page at a later date. Right now, I would like to turn it over to Dr. Michael Kerman, who will be introducing our panel for today's Arts and Sciences Alumni Academy. Michael, please go ahead.
Thank you, and good afternoon, everyone, and thank you for joining us today. My name is Michael Kerman. I graduated from SU in 1973 with a degree in biology. I am a medical oncologist by further training. I held several leadership roles in the pharmaceutical industry before beginning my own consultancy practice. I'm also a member of the College of Arts and Sciences Dean's Advisory Board and previously a member of the Department of Biology Advisory Board. As many of you know, the College of Arts and Sciences is the largest and oldest college at SU and is the home of the liberal arts at SU. Every student at SU takes classes in arts and sciences, and so teaching is a huge part of its mission. A&S is also the driver of the university's research mission, accounting for roughly 1/3 of all research expenditures at the university. And as you will be seeing in today's presentation, our life sciences faculty are world-class researchers who are also committed to undergraduate teaching and research. So today's topic concerns genome editing and the technology known as CRISPR, which stands for Clustered Regularly Interspersed Short Palindromic Repeats, which is truly a mouthful. And these are little pieces of DNA that were discovered in bacteria. But what may even be more important were the genes that were associated with these short pieces of DNA. And these were called CRISPR-associated systems, or Cas. And it turns out that one of them, Cas9, is an endonuclease. And an endonuclease is an enzyme that can snip out pieces of DNA and sew in new pieces of DNA, and therefore can modify a gene. And this work was deemed sufficiently important that it was the subject of this year's Nobel Prize in Chemistry. Some people have viewed this as a sort of a Brave New World or a Frankenstein type of technology, but others see this as the path to curing things like sickle cell and muscular dystrophy. And so right now, I would like to introduce our presenters. Professor Kate Lewis is the chair of the Department of Biology and the Laura J. and L. Douglas Meredith Professor for Teaching Excellence. Dr. Lewis's research studies involves neural circuitry and function, as well as cell fate specification. The knowledge that she and her colleagues gain from her lab can be used to develop more effective treatments for nervous system disorders and tumors, and to facilitate the repair of particular nerves after injury or neurodegenerative disorders. Dr. Lewis received her PhD from the University College in London. Prior to joining SU, Dr. Lewis was a Royal Society University Research Fellow at the University of Cambridge in England, and was also a Kennedy Memorial Scholar at Harvard. Dr. James Hoogland is an associate professor with appointments in the Department of both Chemistry and Biology. His research focuses on the chemistry and biology of protein post-translational modifications linked to diseases such as cancer, cardiovascular disease, and obesity. And understanding these modifications and the enzymes behind them can help us identify new targets for therapeutic intervention and inhibitor development. Dr. Hoogland received his PhD from the University of Chicago. He came to SU in 2010 following an NIH postdoc at the University of Michigan. And finally, Peter Fioramonti is a senior biochemistry major and a research assistant in the lab of Professor Heidi Henley in the Department of Biology. Peter assists in research uncovering intracellular membrane trafficking and signaling mechanisms associated with developmental disorders, such as ciliopathies and cancer progression. And so without further ado, Professor Hoogland, please get us started.
Certainly. Thank you very much for the introduction. Here we go. There we go. Hopefully everybody sees the slides. So welcome this afternoon to our presentation on the CRISPR revolution. And as you can see here, I went and pulled a few journal covers over the years that really genome editing and CRISPR has been recognized as really groundbreaking technology in science for many, many years, with the progression being capped recently by the 2020 Nobel Prize in Chemistry to Emmanuel Charpentier and Jennifer Doudna for the development of a method of genome editing. So what are we talking about here? And really what we're, as so ably was stated in the introduction, CRISPR or CRISPR-Cas9 refers to this clustered, regularly interspaced short palindromic repeats. And don't worry, I'm not going to say that again. This was originally discovered in bacteria as part of their immune system. It's a good time to talk about immune systems. We're all thinking about our own immune system and the COVID vaccines. Thank God for science. But what happens in bacteria is CRISPR-Cas9 is the bacterial way to try to defend the bacteria from invading foreign new genomic information, effectively bacterial viruses. And it involves two different parts. First, as was mentioned, Cas9 is an enzyme that cuts double-stranded DNA. So if you see down here in the gray, we have the two strands of a DNA strand, of a DNA double helix. And Cas9 is, again, that endonuclease, the enzyme that'll cut through both of these strands. Now, the other part of the system, and really what the distinguishing characteristic of CRISPR-Cas9 is the targeting system, this guide RNA shown here in green and in gray. And what that sequence will do is help Cas9 find where it's going to be cutting the DNA at. Now, that really is what makes CRISPR so powerful. If you had to say in a word, what makes CRISPR different than everything that came before, it's adaptability. There are other genome editing technologies that have been around for many, many years. Here are two. If you play around or read in the literature, previous systems like zinc finger nucleases up here or TALEN technology could accomplish the same end goal of cutting DNA, doing the first step to break the DNA double helix on both strands to do genome editing. But in both of those cases, the targeting of the DNA, how do you choose where it gets cut, is very dependent on the structure of the protein itself. The trick to that is it doesn't make it very easy to change the selectivity. So in that case, we have a pair of scissors, but they always cut at the same place. The nice thing about CRISPR-Cas9 is that by changing this guide RNA shown on this slide in orange, this allows us to exquisitely target the cut, the genome editing, two different sections in whatever genome we're targeting, whether it's bacterial, mammalian, etc., to go in and start cutting the DNA to start the genome editing process. That makes this very fast to target to different areas and relatively inexpensive, especially compared to other technologies. So how does this actually work? Again, we're going to have our DNA we're targeting. We have the Cas9 enzyme shown here in orange, the guide RNA shown here as before, and we have two different possibilities. First, it comes in, CRISPR-Cas9 recognizes the DNA and cuts it, and we get what's called a double strand break shown here. Now, our cells can recognize DNA damage and want to repair it, but the challenge is if you cut both strands, there is no templating effect. So if we cut one strand here, you could read from the other strand and repair the DNA. But if you cut both at the same time, there's no template. Now, our cells can repair this, but the problem with non-templated repair is it's very, very messy. It's not very accurate. And so what we can get is deletions of DNA, we can get insertions, and we're going to hear more from Kate, from Professor Lewis in her research about exactly how this comes to pass. But generally speaking, this can lead to loss of gene or protein function. Effectively, you break the gene, whatever you've edited. Another option would be to do basically to provide the DNA a cheat sheet. So here we have that same cut we talked about before, but if you provide a donor template, so this in red would be the sequence of the gene we want, particularly if you're trying to address a genetic disease like sickle cell, like Tay-Sachs disease, for example. You can then, the cell can use this template to do templated repair, which would then hopefully lead to restored or repaired gene, protein, or function, okay? So over here on the left, this would be sort of a gene knockout we would talk about. You basically break the gene you're targeting. On the right, templated repair would give you a way to repair the gene to recode it to the sequence you want. Now, when we think about how this would be applied, particularly therapeutically, we can think about CRISPR-Cas9 being used in two different modes. The first, shown here ex vivo outside the body, has already been accomplished in clinical trials, where particularly for blood-borne diseases, we take blood cells out of the patient, we do the genome editing, then you take the cells that are edited and put them back into the patient. Now, the benefit of this type of treatment where you're able to do it outside the body is we can quality check the cells to make sure that we edited them in the right place and that the editing we wanted led to the desired outcome. This would be used for diseases such as sickle cell anemia, like thalassemia. And as I said, this has already been into clinical trials with some great success stories. The other option, which is a focus of a lot of research right now, is in vivo, so directly in the body. So the idea was we would package up CRISPR, the genome targeting sequences, and Cas9, deliver them into the body, and do gene editing at the site of the cells directly in the body. This is much more difficult for a number of reasons. For example, are we editing all of the diseased cells? Like you're trying to hit your whole body. Right now, the technology isn't efficient enough to pull that off. Also, and we're going to hear again more about this as we hear about Kate and Peter's work, questions about, are the disease cells that are edited all edited in the same way, right? So do you get the same change everywhere? And so these are some of the challenges that have to be addressed as we move forward. Beyond that, we also have to consider that when we edit a single gene, we have to understand how that gene is interacting with all the other proteins, all the other information in the cell. So when we think about genome editing, imagine this Jenga tower here is your genome. What we have to be careful about when we genome edit is realistically right now, you're effectively playing Jenga mostly blindfolded. We know where we're making our edit, but without a really good idea of what's below and above, what proteins, what are the parts of the cell are interacting with that gene we're editing, it could lead to not great outcomes depending on what happens. So it really emphasizes the importance of understanding the context of the gene we're editing in the larger biological system. So beyond sort of those technical challenges, and this is kind of brought up before in the introduction, we also are considering the ethical concerns about human genome editing, which is, these have been around for many, many, many years, basically as long as we've known about the concept of genome editing, but CRISPR-Cas9 and the power of the technology has again brought them to the forefront. And I believe there will be another Academy event later on, or actually I believe in the new year, addressing specifically these concerns. So one issue is, again, there are some diseases like sickle cell, like thalassemia, or universally fatal genetic diseases like Tay-Sachs disease, where you would think that there's a strong argument for applying gene editing technology. But at the same time, we also have to think about the implications of gene editing in humans and what that could mean. Also, a key concept here would be what type of cells or what type of genes are we editing? And you'll see terms of somatic or non-inheritable changes versus germline or inheritable. So a somatic mutation would be something that just affects, say, me. Germline would be inheritable, so it could affect my genetic makeup, but also all of my progeny, all my descendants. And so these are concerns that we have to consider as you think about the technology and the ability to apply CRISPR-Cas9 therapeutically to address human health. So that gives us a little bit of a starting point to consider the application of CRISPR-Cas9. Our next speaker, Professor Lewis, will talk about how we apply CRISPR-Cas9 or how she applies it in her laboratory.
So good afternoon, everybody. So Jimmy just gave you a great introduction to CRISPR and also to how we can hopefully currently and in the future use CRISPR to help with human disease. Peter and I are going to tell you a little bit more about how we use CRISPR in our research and how that's helping us to learn more about biological processes that are also important for human diseases and disorders. So the big picture question that my research investigates is how different types of nerve cells are made and assembled into functional neuronal circuitry as an embryo is growing. But instead of studying the brain, we study the spinal cord. And we do this because the spinal cord is required for movements and also for receiving and processing sensory inputs, so things like pain and touch from the body and the limbs. And if the spinal cord is damaged by injury or disease, then this can obviously result in paralysis. And spinal cord injuries are reasonably prevalent, and for the individuals involved, they can be catastrophic. So our hope is that if we can learn about how spinal cord nerve cells are initially made, we'll have a better chance of being able to grow new nerve cells to treat people whose spinal cords have been damaged by injury or disease. So to do this research, I use zebrafish, and I do that because zebrafish, even though they look very different to us, their genes and their spinal cord nerve cells are very similar to humans. However, unlike humans, they lay eggs, and those eggs are transparent. So it's really easy to look at nerve cells in live zebrafish embryos as they're growing. So here you're looking at some nerve cells that have been labeled with a fluorescent protein called GFP, green fluorescent protein. And here, this is the cell body of the nerve cell. And all nerve cells have this long protrusion that they extend to talk to other cells called an axon. And there are several different types of nerve cells in the spinal cord. And what my lab's really interested in is how these are made in growing embryos. And in particular, what are the genes that instruct cells to make a specific type of nerve cell? So some of you may remember that genes are stretches of DNA that encode for proteins. And so if we want to know what a gene does or what it's important for or what a protein does, then one way that we can do this is we can interfere with the function of the gene, and we call that mutating it. So we can delete the gene completely, also called a knockout, or we can make a change to the gene's DNA so that the protein that it makes no longer works properly. And that's what we use CRISPR for in the lab. So as I said, we can completely cut out the gene, and I'll show you an example later about how we can do this. And then obviously we have no protein, and so we lose the function of that protein and that gene. We can also make a change to the gene in similar ways to what Jimmy was just telling you about, which will mean that we'll have a changed protein. It may have a different shape, it has different properties. That may lead to a complete loss of function of that protein's normal function, or it could lead to its function being changed in some way. So as I said, we're really interested in what genes instruct cells to make a specific type of nerve cell, because then we can use those genes to make stem cells grow particular types of nerve cells for therapeutic purposes. So one key thing that makes nerve cells different from one another is how they talk to other cells, and in particular, what chemical messenger, and we call the chemical messenger a neurotransmitter, they use to talk to other cells. So this is a nerve cell here, and it's talking to this nerve cell down here. This long protrusion, the axon, is stretching out and contacting this second cell. And if we zoom in on this region here, we see what's called the synapse. And what you can see is that this nerve cell at the top is making these red proteins here. They're being packaged here into a vesicle and then released into the space between this cell and the cell it wants to talk to. This cell here then takes up that protein, and that's actually one of the major ways that nerve cells communicate with other cells. And those chemical messengers, those neurotransmitters, they can be either excitatory, in which case they turn this next cell on in the neuronal circuitry, or they can be inhibitory, they turn it off. So to give you an example of a project that looked at this in my lab, we were interested in this particular type of nerve cell in the spinal cord that's called V0V, and these are the only cells in the spinal cord that turn on two particular genes, EVX1 and EVX2. So we wanted to know, what do genes EVX1 and EVX2 do in those cells? So we made mutations in both of them. And so what you're looking at here is a region of the body. So this dotted area here on this schematic of this zebrafish embryo. And the spinal cord in zebrafish runs down the body in the kind of the back side of the body, just like in us. And each little blue oval that you see here is an individual nerve cell in the spinal cord that's making an excitatory chemical messenger or neurotransmitter. WT stands for wild type. That just means these are the normal embryos. And then over here, we have the mutant embryos. And what you can see is that there are far fewer of these blue ovals. So there's far fewer cells in the spinal cord making these excitatory neurotransmitters. But if we look at inhibitory neurotransmitters in contrast, now there are way more of these blue ovals in the mutant compared to the normal or wild type situation. And so that tells us that the genes EVX1 and EVX2 are required for these nerve cells to be excitatory rather than inhibitory. So how do we do this? How do we actually make these CRISPR mutants? So what we do is we take the eggs when they're laid and we inject those eggs or the embryos with the CRISPR reagents that Jimmy was just telling you about. So we use this fine needle and we use pressure just to push those guide RNAs and those other reagents into the embryo. And then we grow those embryos up, and about 3 months later, we get some fish. Now, the CRISPR reagents do not change the DNA in every cell. They just change the DNA in some cells. So some of the cells will prepare the DNA after it's been cut perfectly normally, and others will make mistakes in the repair. And so this fish is mosaic. Some of its cells are completely normal, and some of its cells have one copy of the gene we're interested in that's been mutated, where a mistake has been made. So to identify the mistakes or the mutations that we want, we breed this fish with a normal or wild type fish. And some of the eggs or the sperm cells, depending on what sex it is from this fish, will carry mutations and some won't. So depending on which egg or sperm cell is fertilized or used from the wild type cell, we'll end up with fish 3 more months later. Some of them will be completely normal because they got a normal egg or sperm from this founder fish. Some of them will have one good copy and one mutated copy of our gene of interest if they've got a mutated egg or sperm from this founder fish. So we identify the ones that carry the mutations, we breed them together, and about a quarter of the embryos will have 2 mutated copies of the gene, which means that gene will not be working at all, and we can look and see what the consequences are for the spinal cords in those embryos. So when we're making these mutations, we usually actually design guide RNAs to several different locations in the gene because some of them work better than others. So here you're looking at a schematic of the DNA and the gene, and the green boxes are the parts of the gene, the DNA, that are actually going to encode for the protein. So for those of you who remember kind of your gene structure, these are the exons and in between is an intron. So these black lines here, C, D, E, F, are sites where we design guide RNAs. And when we cut with this guide RNA here at this E site, one of the things that we saw in some of the fish was as we got this truncated protein being made rather than the normal protein, which is shown here in the schematic. So normally the protein looks like this, and we got a much shorter version. And actually the end of it was different amino acids, different building blocks to the normal protein. So why does this happen? So DNA, many of you may remember, is made up of stretches of 4 different nucleotides, A's, T's, G's, and C's. And the way that this gives you the information for what protein should be made is that triplets of nucleotides, groups of 3 nucleotides, tell you what amino acid or protein building block should be used in making the protein. So if we cut the DNA with a double-stranded break, as Jimmy said, that means often it makes mistakes when it's repairing it. So when the cell makes a mistake, if it deletes or adds a multiple of 3 nucleotides, then that's going to mean that you lose an amino acid or you gain an amino acid. But most proteins will still work okay if they're just missing 1 or 2 amino acids or they've gained an extra couple. However, if the cell, when it's repairing that double-stranded break, adds a multiple that's not a multiple of 3, like for example here 4 nucleotides, or deletes 1, 2, 4, 5, 7 nucleotides, then what that means is that this triplet code is going to change, as you can see here. And now, while this is still going to code for the same amino acid, now we're going to get different amino acids. So we're going to get a completely different protein sequence. And very often when this happens, very quickly, a stop codon, a triplet code that tells the cell machinery to stop making the protein, will be encountered, and we'll get a truncated protein. So that's exactly what happened here. When the cell repaired the break in the cell that basically eventually gave rise to this fish, it added 1 base pair. So that put that triplet code out of sync. We say that it caused a frame shift, a shift in the frame. And so then it made several amino acids. It added several amino acids that were incorrect. And it then encountered a stop codon and it was truncated. Now, when we cut the DNA with a guide RNA like this in a particular position, in different fish, we often see very different mutations because cells will repair it wrong, but in different ways they will make mistakes. So this just shows you some other examples of mutants we obtained from this same experiment cut in the same place. And you can see that we got different numbers of nucleotides deleted, and this resulted in different types of proteins. So the other thing we can do is we can actually use two guide RNAs at the same time. So we do a double-stranded cut in two different places. And if we do that in a small frequency of cases, the cell will actually, in trying to repair that, will completely delete all of the sequence between the two guide RNAs and the two cuts. And in that way, we can actually make deletions of whole genes. And we also do that quite a lot in our research. So this is actually really great, because what this means that using CRISPR, as Jimmy was saying, we can design sites in many different genes in many different places. So we can, in our own lab, mutate and investigate pretty much any gene that we're interested in. And we can make them in the lab ourselves relatively cheaply. It just takes a while because, as you saw, we have to grow up 2 different generations of fish before we can really do the experiments with them. Previously, we used to also work with mutants, but we were much more limited. So we had in the past used talons, but we had to pay someone else to help us do it. And that only works for some genes. And in fact, the talons mutant we made, we never worked because we were always getting multiples of 3 nucleotides deleted. So we were never able to kind of get these other deletions that cause the protein to be different. And then otherwise, we were restricted to investigating genes that mutations already existed for. So great thing now is whenever we find a gene that we think is interesting and important, we're able to test our hypothesis about what it does in the spinal cord by mutating it using CRISPR. So CRISPR is really enabling us to identify all of the different genes that make these different nerve cells in the spinal cord that we require for movement and also for sensation in our body and our limbs. And just as a final slide, I say, if you want to hear anything more about this or hear from some of the people who work with me on this, if you just Google Innovation Orange and my name, you'll come up with a short little video that features some of the people in my lab, as well as you'll hear me talk a little bit more about research. And it also talks a little bit about how we use zebrafish in some of the classes that we teach. So thank you. And I'm going to stop sharing and hand over to Peter.
Hi, everybody. My name is Peter Fioramonti, and I'm an undergraduate biochemistry student, and I do research in the Henley Lab. So I'm going to be talking about how I use CRISPR to study Rab11 function in human cells. So oncoviruses are viruses that can cause cancer, and the World Health Organization estimates that these infections cause about 18% of human cancers. So this is what a retrovirus looks like, and it can cause cancer by inserting its viral DNA into the host genome and then disrupting surrounding genes that are important for regulating cell division and proliferation, and genes generally important for preventing cancer. So this might sound really bad, but this actually suggests that 18% of cancer cases could be prevented with vaccination or treated with less toxic antiviral compounds. And now that we're a lot better at developing vaccines in general, this could be a nice thing to do after the coronavirus vaccine. So with researchers who were studying oncoviruses in the 1960s, they identified a family of proteins called the RAS superfamily of GTPases. They discovered these proteins and noticed that there was mutations or abnormal regulation in rats that had these oncoviruses inducing cancer. So you can see these RAS proteins in blue. And the thing that's cool about these proteins is that they can function as molecular switches. They can exist in an on conformation or an off conformation. So this switching mechanism gives it the ability to participate in signaling pathways that regulate cell growth, survival, and program cell death. And since these initial studies that were done in rats in the 60s, they've been studied in humans, and it's been identified that abnormalities in these proteins is observed in 30% of all human tumors. So if we can understand these proteins, we can have a better understanding of healthy cell division and unhealthy cell division. So CRISPR can be used to target specific GTPases. Here is the entire RAS superfamily, and it's made up of subfamilies and hundreds of different proteins. And unlike methods that use GTPase inhibitors, which target groups of these proteins, CRISPR can be used to target a specific protein. So I use CRISPR to study Rab11, which is this GTPase right there. And generally, a better understanding of RAS GTPase function can be used to develop therapies which specifically target malignant cells over healthy cells, and also just provide us with a better understanding of cell division, because our current model of cell division isn't complete. So we use CRISPR to knock out Rab11. So we introduce a cut into the gene and that prevents it from being expressed normally. So this is a Western blot, or a probe to look at the protein concentrations within a cell. And you can see in the normal cells that they have a decent amount of Rab11. And in the cells that were treated with CRISPR to knock out the Rab11 gene, Rab11 isn't present. And then we compare that to another protein to make sure that there weren't off-target effects. And we can see that this GAPDH protein concentrations remain the same. So we specifically decreased the amount of Rab11. And now that we have two cell lines, one that has Rab11 and one that doesn't have Rab11, we study the differences between these two lines to try to understand what Rab11 is important for. So on the top, we have control cells, just normal cells, and we stain the nuclei with DAPI. So you can see in normal cells, they have one round nuclei, and comparing that to the Rab11 knockout cells, you can see this binucleate phenotype. So that means that there's two nuclei in one cell. And this is a sign that cell division is not successful because the two nuclei weren't separated. They just remained in the same cell. So from this, one of the major conclusions we've already made in a paper that was published just this year was that Rab11 is required for cell division. And then my current work that I'm working on this semester involves introducing mutated versions of Rab11 and studying how this affects cell division and centrosome dynamics. So we kind of, on a scale from no Rab11 to normal Rab11, we use these mutant versions to see how they are important for cell division. And that's all I have. I just want to thank everyone in the Henley lab, specifically Dr. Henley and Nikila and all the contributors. And thank you all for your time. And we'll transition to the Q&A.
Thanks, Peter. That was great. And thanks, Professors Lewis and Hoagland as well. So we have about 25 minutes left for some Q&A. And I see some people putting their questions into the chat. That's what I would encourage you to do. If you have a burning desire to ask your question or make your comment verbally, we can do that as well. Go ahead and use the raise hand feature in Zoom, and I will call on you when it's your turn to speak. But I'm going to get things started right now. We had a question submitted earlier by Bob. Bob's question was, CRISPR technology, like many other groundbreaking discoveries, has both great potential for good, as well as the potential for both intentional and unintentional harm. How does SU monitor and discuss bioethical issues that may arise from technologies like CRISPR with the SU research community? Kate, do you want to take that one?
So I can answer that specifically for research using animals like I do that have backbones, so vertebrate animals. So those are very tightly regulated, and any research that we do with a vertebrate has to go to an animal care committee, and they look at the experiments we're planning on doing, and they look at whether there's any potential for harm to be suffered by the animals. But they're also kind of judging whether it's worthwhile using animals for the research as well. And that is a body that consists of other people from the university, but it also has members of the general public on it. And everything we want to do has to be approved by that body before we can actually do it with the animals. More widely, I would say that, I mean, bioethics is something that's really kind of obviously important, and it's also very important that we expose our students to considerations of bioethics. And so we do that with our graduate students through one of the seminar classes that they take. We have some sessions on bioethics where they talk about this, and I think professors bring that into the individual classes as well when they're talking about these, you know, different kind of techniques and technologies that are becoming available. Jimmy, do you have it?
Go ahead, Jim.
Yeah, I would echo. So I don't work with the vertebrate animals. I'm a chemist by training, so I stay a little lower on the organizational chart, one might say. But it is something very important, and we do discuss it in classes. I know we also discuss it a lot with our trainees in the laboratory, involve them in what's involved in putting these protocols together and knowing not just the how of the experiment, but the why, and thinking about the ethical, bioethical implications of it. So it's something that, and I've taught a medicinal chemistry course in the chemistry department over the years, and it's something also we talk quite a bit about not just the how of the medical development, but the ethical and social impact of those. So to put the scientific work into a context in larger society.
Thank you. Okay, we got a question from Mike. Mike said there's several companies that work with CRISPR development. Are these companies able to become commercially profitable? Do you guys know anything about the business of CRISPR? Go ahead.
Which one?
You can go first, Kate.
Okay, so I mean, there are companies. So I told you that we can design CRISPR reagents in my lab and we can use them in my lab, but there are also companies where I could just pay them and they would make me a zebrafish mutant, or they might make somebody that's with a mouse mutant. And I think that some of those, it depends a lot, right, on who has the money to be able to pay for those services and how well an individual company is advertising to the right people. But some of them definitely, I think, are commercially profitable. Whether they all will be, I don't know. And then with any new medical technology, it's all going to depend, I think, on who manages to find something that they can patent that might then become part of a therapy. But I would imagine some of them probably will, and a lot more will probably fall by the wayside.
Right, I would echo. I've gotten the interesting sort of not first person, but next to first person view. One of my former graduate students who did her doctorate here went, Soumya Gangyopati, went and learned CRISPR technology at Harvard Medical School, and now she's working for her second biotech in the Boston area. The one she's working for now is specifically looked at applying CRISPR technology for cardiovascular disease. So they are trying to explore this, and just as Kate said, if I had to guess 10, 15 years out, there will be some therapies. For example, the bloodborne diseases, I think we're going to get there pretty quickly because our sickle cell and thalassemia are already in the clinic for treatments for those diseases. But it's the nature of the biotech that a lot of them do well. A small number do well and usually end up either developing or getting brought into larger companies, or they'll be able to forge the path on their own. The great example I like to say is 10 years ago, I heard these exact same arguments about a little company called Moderna, which I think we all know their name now because of the mRNA vaccine. So it's a nice thing about small biotechs. They do tend to push this technology forward pretty quickly.
Great, thank you. Stu had a question. Do the RAS individually affect respective cancers, or do they do it as groups? And Stu says his 1970s SU biochem doesn't recall if that was known. I don't know, Peter, is that a question for you?
Yeah, I can answer that. So a lot of the RAS proteins are essential for division across cell types, but then the mutations can be specific to a certain kind of cancer. Like a mutation can overlap to multiple types, but with the RABs that I study, it's common to affect neurodegenerative diseases. So there's overlap, but you can kind of specify which kind of disease they're associated with.
Thanks, Peter. Michelle had a question. Are there computer models available to help with CRISPR research that can be used instead of relying on 3 generations of zebrafish? Kate?
So it depends whether you're asking about learning more about CRISPR as a technology, or if you're asking more about if there's things that we could do to answer the questions we want to know about what genes are important for making nerve cells that we use zebrafish for. For the latter, it's kind of really important that we do these experiments in animals. And this comes back to what Jimmy was saying at the beginning about playing Jenga blind. So the issue is that genes in general are turned on in many different cells, and they might do different things in those different cells. But just because they're turned on in a cell doesn't mean they necessarily do something important in that cell. And so the only way we can really test that is to interfere with the function of the gene and see what happens. And we're really interested, as I said today, in the spinal cord, but very often when we look at genes that play an important role in the spinal cord, we find that they play an important role somewhere else as well. So the mutants I was telling you about at the end there, which were in a gene called HMX2 and HMX3, those, when we mutated them, we saw defects in the ear. And so actually the important thing there is that maybe those genes will also be very important for understanding deafness. But we wouldn't necessarily have predicted that if we'd just been trying to computer model it. We really do need to look and see what happens in the organism when we take the function of that gene away.
Thank you. Eric had a question. Is it possible to insert a selection marker into a pre-existing CRISPR-Cas9 plasmid? Who wants that one? Go ahead, Jim.
I would think so. I mean, so especially if we're talking about gene editing at the cell level, which would be where you'd be using plasmids for transfection, most likely. You could put in a selection marker. I've seen some of that technology. More often I've seen, instead of a selection marker, so the selection marker would, commonly can be you put in another gene that's going to allow the cell to survive being in a hostile environment. So if you throw in a toxic chemical, the gene will help it survive. I've also seen quite often people will put fluorescent proteins encoded in these plasmids, so the cells that get transfected light up, and we can tell which ones have it on board. But part of the challenge of CRISPR-Cas9 is it's not enough just to get all the genes into the cell. It's about getting them to assemble and to cut the genome where we want it to. And so there's a subsequent step where, like Kate and Peter talked about verifying where the genome editing is. And so it's a multi-step process to know we got the reagents into the cell where we want them to do their work, but also what is the result of the gene editing itself, and is it the type of modification we need for whatever purpose you want to do.
Thank you. Let's see. Marcia asked, has the Rab11 research been used in animals to change proteins?
Yep, so in the Henley lab, we also do work in zebrafish, specifically zebrafish embryos. So I don't know if we've used CRISPR to knock out Rab11, I'm guessing we have, but we've also used related techniques like optogenetics to disrupt Rab11 localization at specific times. And we've seen that this also results in abnormal cell division and other disease phenotypes.
Thank you. The question from Nikila, how specific is the CRISPR genome editing system, and how would you account for off-target effects?
So, and I tried to kind of give a sense of this at the end of my talk, it's not very specific, right? So if you do, so Jimmy presented the two different kind of major approaches. The one where you just cut the DNA and you let the cell try and repair, the cell can repair it in many different ways. And so it's a little bit random what you get. So you have to sort through what you get and find what you want. And that's why the therapies where you're taking cells out of the body and manipulating them work quite well at the moment, because you can then look at the cells and check that you got the change that you want, and that it's only those cells that you'd put back into the body. With the fish, we have to actually screen through lots and lots of fish and look at that particular region of the DNA and check what type of change is made and whether it's what we want or not. Now, you can also, the other method, as Jimmy was saying, is you can provide a target, and then hopefully, at least some of the time, the cell will use that target as a template to make the repair. But at the moment, that's not very efficient. So while that may happen some of the time, at least in experiments I know of using zebrafish, it might be 1% of the time. So maybe 1 in 100 of the cells gets repaired in the way that you want, and the other 99 might not. And that's why it's really difficult at the moment, I think, to treat directly into like human embryos or into, humans for a lot of these diseases, because you can't know for certain that you're always going to get the changes that you want to have happen.
AG had a question. Any update on the use of CRISPR in CAR T therapy? Also, how do you control for off-target modification? Jimmy, you got something on that?
Yeah. So I know when I was doing some of the preparation for the background, so yeah, CRISPR is being applied a lot in CAR T therapy. So the idea of trying to reprogram immune cells. So this is for immunotherapy targeting cancer. Can we target immune cells to attack the cancer inside the body itself? So it's being used quite a bit. I wasn't able to find if it's gone significantly forward in clinical trials. So I don't know if we're to the point that it's actually gotten back into patients yet, or how far along that's gone. But there is a lot of optimism in the field that is one way to hopefully rapidly reprogram these immune cells to target different cancers and to use them for immunotherapy. And then any control for off-target modification, like Kate said, that's not even much more than $64,000 question right now. And CRISPR technology is, how do we control both where, when you get to a larger context, how well can we control where it's being modified, where we're doing the gene modifying, what kind of modification we see. So these are some of the real key challenges facing the field right now.
Great, thank you. Question from Abrar. Peter, are binucleate cells the only outcome of knocking out Rab11? Do you see other effects that are cancer related?
Yep, yeah, we actually do. So when we knock out Rab11, we also observe centrosome abnormalities. And the centrosome is the organelle that's really important for regulating cell division. And these are abnormalities in the structure and the number of centrosomes that we observe are similar to the ones observed in certain types of colon and prostate cancer. So it looks like there is a pretty close connection with the RABs and cancer cell division.
Thank you. Nicole asked, do you know of any current methodologies that can increase the efficacy of gene editing across all cells with the Cas9 system? Who wants to take that one? Nobody.
I don't.
Okay. All right. Jamie?
I'll throw a little bit out there. I did see some literature that, so we talked about Cas9, and as Michael noted in the introduction, that it's, Cas is CRISPR associated, I forget what the S was. Well, I'm a chemist, I can't remember the names. Cas9 was one of the major journeys, but there are other Cas proteins. So I've seen, I think it's Cas12, Cas13. There is some exploration using different enzymes that may be more efficient at cutting. Also, related to that, one of the target challenges is delivery. One thing about Cas9 is, as proteins go, Cas9 is a pretty big protein. It's about, I think, about 150 kilodaltons. Basically, it's very, very large. For example, it's about 5 times larger than RAS. And so part of the practical challenge is to take all the parts you need for CRISPR-Cas9 and officially get it into whatever cell you want to edit. And so there's a lot of development there at the delivery side of how do we get the reagents to where they need to be to have their effect. So if we can improve delivery, it can also improve outcome.
I'll add something to that. So that made me think of something. So one of the things that does make a difference with experiments we do where we're injecting those reagents into a zebrafish egg is whether we use Cas9 RNA, in which case the cell has to actually take that RNA and turn it into protein, and there's a bit of a delay, or whether we just give it the protein to start with. And if we want to try and make changes that will affect as many cells in that fish as it's growing up as possible, it's better if we give it the protein, because then it starts to actually cut quicker, which means that it starts to make the repairs at an earlier stage when there's fewer cells. So if it waits and it's not really repairing cells until we've maybe got 32 cells, because we've had the cell division start, then all of those 32 cells might get repaired differently. But if that repair happens maybe when it's only 2 cells, then at least half of the embryo is going to have the same repair and the other half would have the other repair. So definitely by playing with things like that, like whether you're adding protein or whether you're adding RNA, that can sometimes make a difference.
Terrific, thank you. I think that is it. I don't see any other questions coming in. So I think we may be ready to wrap up. Let me tell you first, though, that if you're interested in this topic, and we got into this a little bit today, we're going to be doing a companion program in mid-January on the ethics of CRISPR. And we're going to have a couple of our philosophy professors come in and talk about all the different ethical issues that this technology raises. So keep an eye out for an invitation to that coming out soon. The other thing I wanted to mention is if you would like to support the great work being done by these faculty members and the students like the ones you met today, I would encourage you to make a gift to the College of Arts and Sciences. You can direct your gift to one of the departments you heard from today or to general support of the college. So to do that, go to giving.syr.edu. Gifts of any size are appreciated. Thank you. And with that, I want to say thank you to Professors Hoagland and Lewis and to Peter and to Dr. Kerman. We appreciate you taking time. That was really fascinating and really interesting. So thanks a lot. And thanks everybody for joining. We'll see you next time.